Hi everyone, welcome back. So now we're going to talk about the shape and arrangement that bacterial cells can find themselves in. So what they can look like, particularly when we put them on a slide, like when we're going to do gram staining. So bacterial cells come in a lot of different shapes. A huge amount of variety is possible for bacterial cells. Despite them being relatively simple in appearance, they do have a lot of variation. So the ones that we see most often are the coccoid and bacilloid cells. Like in our lab, most of our species are going to fall within this category, coccoid, bacilloid. So coccoid means spherical, it means circular, whereas bacilloid means rod-shaped, okay? You can also have a coccobacilloid species, which is essentially like it's rod-shaped but very, very short. You can also have spiral-shaped bacteria that come in a few different forms. We have spirillum, which are shorter S-shape type cells, and then spirochetes that are much longer and resemble corkscrews as they move, and they will actually kind of spin as they swim through an environment, which is pretty interesting to see. Vibrio are mainly comma-shaped bacteria. And then pleomorphic species are bacteria that have no real defined structure. They don't have a consistent shape and exhibit a huge amount of variation. These pleomorphic species, super variable, and the reason behind that, a lot of the time, not all the time, but a lot of the time, is because the species that are pleomorphic do not have peptidoglycan cell walls. Like I mentioned in a previous section, most bacteria have these cell walls, have gram-positive or gram-negative cell wall structures. But there are some species that do not, that don't have peptidoglycan in their cell walls. And as a result, they don't have that kind of structure, the rigid shape and consistency that a lot of other bacterial species have. So there's a couple representatives that fall into this category. We have Mycoplasma, which is a whole genus of bacteria that do not have cell walls and are all pleomorphic. And then we have the L-form bacteria. That a lot of these are variations of existing bacteria, like strains or distinct species that emerge and lose their cell wall, which is pretty interesting. There's a few species that fall into this category, like some bacillus, which normally have a cell wall, but there are representatives that are L-form that don't. Now, these species that don't have cell walls, there's not very many of them. But the ones that do exist can be really medically important, very medically important, because they can cause disease, some of them, and as they do, they're a bit harder for us to deal with because some of the antibiotics that, for example, target a bacterial cell wall, well, they can't really hurt something that doesn't have a cell wall. So there's some major issues that arise from that. Now, bacteria. These bacteria all reproduce asexually, right? Mainly through dividing directly into two, one cell splitting down the middle in some way. We call that binary fission. Bacterial cells can do binary fission, and the way that they do that fission, either how they split, in what direction do they split, can influence how the cells are arranged as they form on a surface. Okay, so essentially the patterns of how cells clump together as they reproduce and spread. And it also can have something to do with the mobility of those bacteria, like how they actually get around. Species that are highly motile sometimes don't stick in one place after the cells divide, and as a result, there isn't really an arrangement that arises. But for the species that do have an arrangement, we have some common ones that we can see. So in bacilloid cells, we can see singles, which means the cells are scattered and have no distinct arrangement. They could be diplo, which means that they occur in linked pairs. They are strepto, which means that they occur in longer chains, or they could occur in palisades, where the cells are stacked side to side instead of long ways, sort of like a package of hot dogs rather than like a chain of sausage links. And then for coccoid cells, we can also have diplo. You can also have single arrangements as well. So you have diplo as linked pairs. You can have strepto as longer chains. You could also have tetrads, which are sets of 4. You could have sarkinae, or sarkina as a singular, which is like a cube arrangement. And then you could also be staphylo, which means that you clump in large irregular patches. Now, when bacteria start to grow together, there are some interesting survival strategies that can actually emerge here. Some bacteria will do certain strategies and others will do different strategies. A major, very important strategy that some bacteria do, is forming a biofilm. So, on their own, single bacterial cells are very vulnerable. These are planktonic, meaning they're free-living, they move through the environment, or maybe they don't have the ability of movement. They can be pushed or carried with currents through the environment. They're on their own. And as a result, there's a lot of vulnerability that they have. You put them at risk of a variety of sources of mortality. And bacteria, still have these major defenses they do to survive these various sources of mortality under the right conditions. And one of those is a biofilm. These are essentially a communal structure that bacteria can form and grow together, both with members of their own species, and even in some cases with members of other species. In a biofilm, bacteria are protected from predators. They are able to hold and control a surface they've colonized, which means they keep on maintaining access to some very important nutrients and also, the valuable space that they have. And it's also the maintenance of a safe place for the cells to actually reproduce and create more of these bacteria. So, motile bacteria, when they go out and about, and in their environment, their flagella carry them around, they can move around and are often looking for signals that indicate a safe place for them to, establish, set up roots, basically, that they can claim a new home. So these biofilm-forming species, they often have a glycocalyx, or like some other structure that helps them adhere to a surface like pili or fimbriae. Whatever the strategy is they use that strategy to attach to the surface. And then once they do, they begin the arduous process of dividing, making more and more and more of themselves, often relatively quickly. They do this especially because in these small numbers, they're very vulnerable both to pathogens, to predators, to environmental conditions. So it is very much the case that bacteria, despite being, species that we often associate with disease, they themselves are capable of catching like a viral infection and things like that. Once these bacteria have established a colony on a surface, they lose their motility if they had it, meaning that they literally, they will destroy or throw away their flagella. They fall off. And as the colony begins to grow, they're going to be producing this extracellular matrix. This is a collection of substances, these extracellular polymeric substances, that they secrete and form like a thick protective netting, essentially, that covers them and holds them all in place and overall protects them. Now, nutrients can still pass through this biofilm and these protective proteins, and at the same time, this matrix can be very protective. So nutrients pass through, but other things that can be more destructive, like certain chemicals, like sterilizing compounds, some drugs like antibiotics, they are often stopped by the tough protective matrix. Now, like I mentioned before, biofilms can be a community affair, not necessarily exclusive to one species. So cells are actively signaling in their environment to other bacteria. Essentially, they're putting up signs that say, hey, there's space here, come live with us. And they're actively recruiting for their biofilm. And they do this not necessarily because they're truly altruistic and they're like, oh, we have to look out for our neighbors, but because there's strength in numbers. The more bacteria that they have on a surface, the less likely a predator that comes by is going to grab any particular cell. And so, it's sort of like the protection that like a herd of zebras gets from the herd itself, if that makes sense. Especially large biofilms, as they get to a certain size, they're going to get big enough that they actually break apart eventually and begin to release bacteria. This biofilm then becomes like a source population where new bacteria can be grown and created in this very protective environment like a cradle, and then be released out into a much more harsh environment. And so from here, bacteria can be released and become planktonic again, regrowing their flagella and then using it to swim into the environment to probably go and find another place to start a new biofilm. And this cycle can start all over again. Now, a biofilm is something that is very medically relevant. It occurs on both biotic and abiotic surfaces. So it's not just like on, a rock, but also on, for example, your teeth. These things can be thick to the point where we often don't even need the use of a microscope to see them. Case in point, the plaque on your teeth. So these medical biofilms, or dental biofilms in the case that I'm talking about, their main methods of destruction, the ones that we want to get rid of them, like how we want to get rid of them, well, we kind of have to get creative because, again, that extracellular matrix, it's really resistant. It's really tough, and it's very selectively permeable. Some things, like specific medications, are not going to be able to penetrate that matrix very well. So we have to utilize sometimes physical methods of removal, like brushing and scraping, and also like chemical methods of removal, like specific compounds that attack the extracellular matrix, like those polymers that make up those structures. And for the latter, there's not really a ton of good options out there. This is why we don't really do chemical treatments to remove biofilms on our teeth. We just brush our teeth, right? We go to the dentist and they take these picks and scrape all the dental plaque off. Most disinfectants and antibiotics really are effective against planktonic bacteria, really are not very effective against these biofilms. So another really important issue with biofilms comes in the form of contamination to implants. So in medical settings, we sometimes have to implant certain structures, like, for example, a pacemaker or a joint replacement or supports for a broken bone. These are things that we often have to do for medical intervention, but these are things that can also carry with them bacteria, and biofilms can establish on these structures. And if they do, this is a major problem, because now you have something that is inside the body that is resistant to a lot of our immune defenses. Likewise, it's very hard for our immune system to attack these biofilms. B, very resistant against antibiotics. So when we develop a bacterial infection, treating it with antibiotics won't necessarily make it go away. And C, it is a constant source of bacterial cells that begin to arise from this healthy biofilm that can continue to reinfect and reinfect a person who received this implant. So this is a major problem. This can even happen with species that are, typically non-disease causing. Opportunistic pathogens, once again, can become disease causing if they were on an implant and then brought in, and now you have this biofilm problem and things like that. So it's super important to keep our medical implants sterile before surgery actually happens, not just because you might introduce a new infection to a person, but that you might introduce a biofilm to an otherwise sterile part of the human body. Now, there are other mechanisms that bacteria can use to survive this environmental hardship that I was talking about, including things like antibiotics. One of the important things that some bacteria, not all, is actually a much smaller group of bacteria that they can do, is form an endospore. This is essentially a really resilient shell of bacterial DNA and a little bit of cytoplasm and a little bit of, other basic resources, like super, super basic things and specific proteins. And so this is something that the cell does when it becomes stressed, when it is experiencing some kind of environmental stress, and it thinks it kind of has this decision-making process where it makes the decision that it needs to survive, even if it must go inert for a long time. So the endospore, when it forms, it's essentially an inert. You could think about it kind of like it's hibernating kind of structure for the bacterium, where the bacterium ceases to function as a healthy, what we call a vegetative cell, and it restricts itself to only be this little seed, like a little bacterial seed, that under the right conditions, it can germinate and emerge and become a big vegetative cell once again, but only if the conditions are right. So these endospores, okay, when they form, they're composed of some important structures. Their shell is really resilient. It's composed of many membranes and a thick outer coat. So this is like a really complex structure that begins to form, and it's so resilient that it can help them survive not just really intense amounts of antibiotics, but also like high heat, strong acidity, unusual conditions that would normally kill virtually anything else. These structures, these endospores, can often survive those conditions. So when these spores form, it kind of follows this pattern. There's a bit of variation here depending on the species, but in general, this is the cycle. You start with a vegetative cell. This is the active cell, a normal, healthy cell that's going about its business, doing bacteria things. When it gets the signal, the internal signal that it needs to enter an endospore, it's going to first create a wall, a septum, that emerges between the genome, which will be somewhere in this small space on this in this diagram, and the rest of the cell. Essentially, it begins to cordon off its own not nuclear. Sorry, they don't have nuclei. Their own like nucleoid genome. Once that nucleoid genome has been sequestered like this, it's been kind of separated, it will begin to form a little capsule in here. That septum is gonna wrap itself around that nucleoid region. Then eventually it's gonna fully separate from the rest of the cell. You get this thing that we call a forespore, right? It's like a not fully formed endospore at this point. Over time, this forespore will continue to shrink. It will concentrate, and a protein coat is going to form around the outer layer. Eventually, this spore will exist inside of an otherwise inert cell. All the DNA is inside of that spore, and there's nothing in this outer cell. And so it just kind of ceases to maintain itself. all of the instructions for how to make all the stuff that a cell needs to survive, well, it's all packed away. And so that the cell itself is no longer doing that job. Eventually, that cell is going to lyse. It's going to break apart and spill all of its contents, including that little endospore now, into the environment. This endospore is going to go about, it's not alive, can't do any of the things that a normal vegetative cell can do at this point, but it will keep on going about, being moved around with the currents, or with just like the movement of things in the environment. And only when the environment changes, becomes more habitable, is it going to sprout from that spore. It's going to germinate. So this whole process is something that we call sporulation. Okay, the process of endospore formation. And by the end of it, there is no vegetative cell. Only the endospore remains. Like I mentioned before, it will exist in the environment until it senses that the environment has changed and has become more habitable. After which point, it's going to germinate and sprout like a seed into a new vegetative cell. A lot of bacteria, not all, can produce endospores. This is something that that only a small group can do. Mainly, it's only in gram-positive bacteria. Okay, so there's a lot of species that are actually really like medically important that form endospores, but again, there are lots of other ones that do that are not, major causes of disease. But the ones that we should definitely talk about are anthrax. This is Bacillus anthracis, tetanus, Clostridium tetani. Also, another species not on this list, but very medically important is the one that causes botulism, Clostridium botulinum. And then C. diff, which I think a lot of us are probably quite familiar with. This is Clostridium difficile. You'll notice that most of these belong to, really two groups. We have Bacillus and Clostridium. These are two genera of bacteria that are pretty prolific when it comes to endospore formation. And remember, this is really something that we see only in some gram-positives. There are some gram-negatives that we think might be able to do this, but a lot of evidence continues to arise that says, these things that we thought formed endospores do something else. So really, this is a gram-positive characteristic, and only a gram-positive characteristic that only some species have. Most others do not. So endospores, in that state, they're highly resistant. They can survive most antibiotics and most forms of sterilization, certainly the vast majority of chemical sterilization. And they can even survive pretty high heat. This is something that we actually have to employ like excessively high heat, or like moist heat methods, like a pressure cooker kind of method that involves this very penetrative kind of heat to actually eradicate them. This is something that we do in like a medical and a hospital setting where we will be using like an autoclave. And we'll talk more about that in a future unit for sure. But in this state, it's really interesting. These structures, these endospores, can survive an incredibly long time. In some cases, upwards of 10,000 years, which can be pretty significant, pretty concerning that, in some environments there can be endospores that formed and then lie in wait for the environment to change and become better. But that's thankfully not something that we really encounter as a major threat on a day-to-day basis for us. But this is certainly a very important aspect. So make sure that you review the process of endospore formation.